Ben has a bachelor's in chemical engineering from University of Arkansas and a PhD in chemical engineering and biophysics from Arizona State University. He served as an assistant research professor for 7 years in the Center for Metabolic Biology at Arizona State before joining Berkeley Lab in 2009. And Ben's really interested in understanding complex, dynamic metabolic networks in environmental context. So he's going to talk to us today about his simile tool.
I didn't realize I'd been here that long. Good times. All right, so I'm giving a short talk today on a new tool we've developed here called Simile, and basically try to tell you a little bit about it. And thank goodness Ming and Trent and everybody's gone before me, so we kind of warmed everybody up. So I'm going to give a little, very brief, one-slide primer on metabolomics to reinforce what you've already heard today. This is how we identify molecules in metabolomics, which is an inexact science, but we run a lot of standards. Trent said 4,000, mine's an older slide, says 3,000. We've run a lot of standards at the JGI, and it is thousands. I mean, we're running more constantly, it's growing. And we achieve identification by comparing their retention time, their M/Z, to make what's called an extracted ion chromatogram. And then we measure what we call their fragmentation spectra, or nowadays it's safer to just say fragmentation data, because fragmentation spectra can be very complex. And we compare that to a standard. So if you get this match in your experiment, you typically would say that that's what your compound is. It's still not perfect, but this is the way the science has moved forward. And as Trent mentioned, we've deposited all of our reference data in GNPS to make it a public resource, and this is being used by the community and has, I think, been really a good thing for everybody. Maybe the good karma also. So now kind of the flip side of that coin, this is more Ming would have been talking about, is the scoring. You often want to understand something about similarity. You don't get an exact match. You get a match that's close to exact. Maybe it has a few extra peaks, the ratios aren't quite right, something like that. So your rows are various compound structures, and you get some list of scores from very low scores to very high scores. And you want to just pick the top score, right? Okay, it's this, it's the top score. But this is some menaquinone from a plant. You know, a little further down the list is a different one from a microbe. And maybe you were growing that exact microbe. So you might think, oh, it's probably that one with a score half as big. So we need a science to be able to explore these similarities. And this is what GNPS has let the public do, is explore these complex measurements in a pretty easy way. So to do these spectral similarities really imply structural similarity. In this case, they do. So here's a cartoon of that to just, again, reinforce what's already been said today. Imagine 4 hypothetical structures, A, B, C, and D. And they're made of these little building blocks of the different colors and shapes, which have different masses. And you can see A is different than B by this one variable mass that weighs 2, and it's on a very specific spot. If I just line up their ions, but this x-axis is mass, and these bars represent a theoretical spectra, you don't get a very good alignment, right? Because it weighs a different amount. You know, the yellow parts line up, the blue parts line up, so you get 2. A versus B has 2 pairs. But if I shift the masses of molecule A to B by their parent masses, you get more ions lined. You get 3. So another ion lines up because of this shifting amount is accounted for in the shift. And that's exactly how we look for spectral similarity as a proxy for structural similarity, is we find something that has a modified mass on one part of a molecule. Now, if we go into C and D, there's multiple modifications on multiple parts of the molecule. You kind of see the alignment falls off the table. It doesn't work as well when you shift or you don't shift. So the scoring isn't as reliable when you have multiple modifications on multiple parts of the molecule. So this is the problem that Simile set out try to tackle and see if we could be successful at solving this problem. Was not requiring a single shift, but allowing more complicated relationships between pairs of spectra. Let me skip through all the animations that I did not use. Okay, this slide has also many animations that I will probably not use. The GNPS is the most widely used tool for doing this. There's another publication called NIST Hybrid Search. And the NIST hybrid search, again, shifts by the parent mass, and they allow you to find related structures. So you can pair up these related structures using this algorithm. That's very similar to what Ming described. And in their abstract, it says what I just showed you with the cartoon. It says this doesn't work if you have multiple differences on multiple parts of a molecule. The association can't be found very reliably. You need to have your difference on one part of a molecule, and it needs to be one difference to work reliably. And then, of course, you've all just heard about the GNPS tool. Their help page says the same thing. If you go to the GNPS documentation, which is vast and very helpful, you will find this is the first sentence in the multiple modifications heading, that if there's two distinct structural modifications, it's not going to work as well. It does work sometimes, though, because of cryptic effects of fragmentation spectra. So here came in the simile tool. So this was inspired by protein sequence alignment. And the idea is, given two spectra, the spectra on the positive axis and the spectra on the negative axis, we're not just going to shift them by one number. We're going to try to line up all the M over Z's from spectrum 1 with all the M over Z's in spectrum 2. And you can go read about it in this jumbled up slide modification, but it was published in Nature Communications recently. And I'll tell you a little bit about it, but it really is high level. So, in protein sequences, you have a substitution matrix that's fixed. You can use the same protein substitution matrix for this pair of proteins, for another pair of proteins, for another pair of proteins, for another pair of proteins. You can think of it as a universal substitution matrix. In fragmentation spectra, we don't know that universal substitution language. We don't have a universal substitution matrix. So what we make is called a pair-specific similarity matrix. So every pair of spectra that come along get their own similarity matrix, unlike protein sequence alignment. But we can still calculate that, and we calculate it by looking at the frequency of M over Z differences in one spectrum and the frequency of M over Z differences in another spectrum, and then apply a small mathematical trick to find the simplest, but yet maximizing the meaning of the relationship of these different M over Z pairs to find correlated or similar MZ pairs in your spectrum. And those would have this high intensity. And then very similar to how you'd use the substitution matrix for protein sequence alignment, you can use a dynamic programming algorithm to find a path through these high similar scores of ions. And you can also calculate a p-value by having a random resampling of this spectra to see how significant this score and path is. So this is a p-value for an alignment. So the ticket to publishing is benchmark, 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 your tool. And this was a heroic effort that I could not have done without Ming. Tremendous help from Ming. This was very difficult to do. The running 5 other algorithms against simile. So the idea here is this purple line is the original distribution of what we call MCS, or maximum common substructure, of hundreds of millions of pairs of spectra. So if you just grab any two spectra, you'll find that the molecules behind the scenes have a maximum common substructure. This is their structure now, not their spectra, of about 0.25. And then these are widely used algorithms like GNPS, modified cosine, simile, the CSS algorithm. All of these algorithms push that distribution to the right. So they find more pairs of more pairs of molecules that are structurally similar. They have a higher MCS. And if you go out to an MCS of 0.8 or greater than 0.7, I have in red here, these are molecules that are almost identical. So one has a hydroxyl group, the other does not. Other than that, this giant molecule is the same. That's what a 0.7 would look like. This is literally aligning two structures on top of each other. But if you look at the raw count of what simile can produce versus the others, simile gives 360% more pairs of molecules. So it just allows you to associate more things together without sacrificing the quality of what's being associated. We did this for positive and negative ionization mode, which is a issue related to the instrumentation. And a very hypothetical test that I find to be the most meaningful, where you grab random flavonoids. So these are little molecules that plants love to make, and random indoles, which look nothing like flavonoids. You would hope all the flavonoids connect to the flavonoid spectra, and all the indole spectra connects to all the indole spectra. But just because of how molecules ionize, that doesn't really happen. When they fragment, there's no law that says when you shift by their precursor mass, they're going to line up. But simile was actually able to almost perfectly do this for the flavonoid case. So finding all the flavonoids connected to all the flavonoids, and it was able to find most of the indoles connected to most of the indoles. So this was a really good, comfortable feeling from that. And as Trent showed, this was able to take one of the most well-studied cosine-based networks in the world from the GNPS original, one of the GNPS original manuscripts in Nature Biotechnology. And we were able to find something new in this well-studied network. So this other peptide-like natural product was able to be found by simile, and it had a near zero cosine similarity score, whereas the simile score was sky high. So that was comforting. And then here at the JGI, we've been using this to try to find related secondary metabolites that this Andrew Osborne, a former postdoc here, just knew they were from the same class of compounds. And he was unable to build a molecular network for them using cosine-based scoring, but the simile scoring linked up the ones that he knew were from the same class of molecules, the same type of BGC. So being a short talk, I now give you my conclusions and acknowledgements. The first author of the simile paper, Daniel Treen, he is absolutely just ate and slept and breathed and everything simile for a long time and pulled this whole thing together. This is a tool that's immediately useful. You can access it on the GNPS Similarity Hub, which is a very useful tool for comparing algorithms. And it's a new scoring framework that might let you find new things in your sample. And that's all I have. I'd like to thank the JGI and Enigma and NERSC for all the funding and support.